Hello everybody, welcome to another session of YouTube Live Standard 9 Science and Technology Part 2. I am Mrs. Priya Mohorkar from SV Union High School and Junior College, Pune, Maharashtra, India. We are going to continue with lesson number 17, Introduction to Biotechnology. We have already started with this chapter. We have seen 3 sessions in which we have discussed few topics of this chapter. So let us continue with the same chapter, lesson number 17, Introduction to Biotechnology. But before we start with a new topic today, new subtopic today, we are going to have a look at whatever we have learned in the last 3 lessons. Okay, let us just brief so that you can remember whatever was done earlier in the early sessions. Now, lesson number 17, Introduction to Biotechnology, has 4 different subtopics. One is on tissues, tissue culture, agritourism, and agribusiness, isn't it? Now we have learned the subtopics tissues, both animal tissues as well as plant tissues, and we also learned about tissue culture, which is a method of biotechnology. And today we are going to start with agritourism and continue with agribusiness. Okay, so let's recall before starting with the new subtopic. So we learned about the tissues earlier. We know a group of cells which have the same origin, same structure, and same function is called a tissue. All the functions of the body occur with full efficiency due to the specific organization of these cells and their division of work. Okay. Now, we also saw that there are 2 basic types of tissues. They are the simple tissues, which are made up of only one type of cells, and the other one is the complex tissues, which are made up of 2 or more than 2 types of tissues. Okay, we saw various animal tissues. We saw what are animal tissues and how they form organs first, but we learned in brief about the classification of animal tissues, how the animal tissues are categorized in 4 different types, of which the epithelial tissue is a simple tissue, followed by the complex type of tissues, the muscular tissue, the connective tissue, and the nervous tissue. Let me take the pen, please, for you. Right, so the epithelial tissue, we learned that it is a simple type of animal tissue, consists of only same type of cells, one type of cells. Okay, but still there are different types of this epithelial tissue. They are the simple squamous epithelium, stratified squamous epithelium, columnar epithelium, ciliated columnar, cuboidal, and glandular epithelium. So we learned these basic 6 types of epithelial tissues. Next, we learned about the muscular tissue, which is responsible for movement in the organism. Now this muscular tissue, we learned 3 types of muscular tissues, which are striated muscles, the non-striated muscle or muscular tissue, and the cardiac muscular tissue forming the muscles. Okay, whereas the next type of animal tissues we learned was the connective tissue. The connective tissue, of course, are also of various types. We know that they connect the different organs of our body, keep them together, and they are categorized into different types. They are the areolar tissue, the adipose tissue, the fibrous tissue, the cartilage, the bone, and the fluid connective tissue, which are the blood and the lymph. Okay, right. And the last type of animal tissue we learned the nervous tissue or the neural tissue. The nervous tissue, of course, because of which we all can respond to the stimulus from the surroundings, of course, and internal stimulus also. Whereas this nervous tissue are of two different types. We learned here they are the neurons and the neuroglia. Okay, right. So let us talk about the next session where we learned about the plant tissues. Right, we have already seen briefed in the last session about the plant tissues. So this is a schematic diagram that shows the classification or categorization of the plant tissues. Now, plant tissues are basically are of two types. One is the meristematic tissue and the other one is the permanent tissue. Now, meristematic tissue actually are those tissues which help in growth of the organism, isn't it? That is, they keep on dividing and dividing quietly but very rapidly, and these help in the growth of the organism, growth of the plants, right? And that is why plants show unlimited growth. Whereas the permanent tissues are those tissues which are formed from the meristematic tissue by certain modifications. We call that process as differentiation. Now, after differentiation, the tissue actually attain a permanent location, and they undergo changes and modify themselves according to their functions. Whichever function they got to perform, they modify accordingly, okay, by the process of differentiation. Now, further, some tissues may further modify, and we call it as redifferentiation, where they become so complex in and so different in structure because they have to perform certain specific, very specific functions. Whereas certain tissues may undergo dedifferentiation, become very simple type of tissues if necessary. Okay, so the process of differentiation may lead to redifferentiation or sometimes dedifferentiation. But whatever it is, the tissues formed from the meristematic tissues by simple cell divisions actually undergo changes to form the permanent tissues. These permanent tissues are said to lose the ability to divide further because they undergo differentiation. Okay, now let us talk about the meristematic tissue. Meristematic tissues are of three different types based on their location. They are classified into three basic types. They are the apical meristem. You can see here, it has been written down here. Apical meristem, the lateral meristem, and the intercalary meristem. Apical meristem are found at the tips of the plant parts. They are the stem, roots, tips of branches, tips of leaves, and so on, of course, and helps in vertical growth. Whereas the lateral meristem actually is found along with the vascular tissue, the vascular bundles now, and they are found on the sides of the plant parts. Okay, lateral refers to the side. Now, actually, lateral also refers to the increase in thickness, increase in the girth of the plant part. For example, if you take stem increases in its size horizontally. If I say horizontally, it will not be right. Anyway, the stem undergoes thickness and increase in the girth of the stem and causes a thick and strong, large stem, which we term it as trunk later on. Okay, so lateral meristems actually help in increasing the girth of the stem, whereas intercalary meristems are found at the base of the nodes. Nodes are the regions where leaves and branches are produced, right? So even flowers are produced at their axils, at the leaf axils, axil of the leaf. So these meristem help in the production of lateral branches, leaves. Okay, of course, they are found at the base of the nodes or just above the nodes in that particular region only. Now, these are the three different types of meristematic tissue. Let us talk about the permanent tissue now. The permanent tissue actually are of basic two types. They are the simple tissues and the complex tissues. The simple tissues are made up of only one type of tissues. only one type of cells, whereas the complex tissues are made up of two or more than two types of cells, right? So let us talk about these simple tissues. Simple tissues are basically three types, categorized into parenchyma, collenchyma and sclerenchyma. Parenchyma are the basic and the most simple type of tissues, okay? And basically their function is storage of food material, and they fill in all the spaces wherever necessary. Whereas collenchyma are slightly differentiated from parenchyma or from the meristematic tissues, wherein they start depositing cell wall material at the corners of the plant cells. Okay, so they have thickened corners in between the cells. That is where they become more strong. They give strength to the whole plant and also give mechanical support to the plant. Whereas the sclerenchyma tissues are dead tissues, okay? They actually start depositing the cell wall material as soon as they start forming the basic cell wall, which is made up of cellulose. Along with it, there are lignin and suberin type of cell walls that keeps on depositing on in the cell wall region, making the cells very thick, as well as slowly they lose their cytoplasm as well as nucleus and become dead cells. So these sclerenchyma tissues are all dead cells. And there are two types of sclerenchyma. One is sclerenchyma fibers, which are long elongated cells having tapering ends. They are dead, of course, whereas and they give mechanical support to the plant. Whereas the other type are called as scleroids or stone cells, which are really stone cells. They have very small lumen inside, and they are made up of very thickened cell walls made up of lignin and suberin, of course, with the basic cell wall material like cellulose, hemicellulose, etc. Along with it, the secondary cell wall materials are found in large quantities. They get deposited in large quantities, making the cells very hard, very rigid, like stone. So they are called as stone cells, also termed as scleroids. So these are the three different types of simple tissues. Now, parenchyma tissue sometimes needs to undergo modifications according to the specific functions. Like parenchyma, the chloroplast in the parenchyma starts dividing and redividing and produces green colored, with green colored pigments called chlorophyll. As they divide, the cells start performing the specific function of photosynthesis. Now, that is why, because there are many chloroplasts in it, the cells itself, the tissue itself is called as chlorenchyma. Chlorenchyma tissues with many chloroplasts in it. They look green, and they are found in, of course, the green parts of the plant, which are the leaves of the plant, okay, mainly the leaves. So you see the leaves green in color, isn't it? Right. Now, not only that, in some plants, the parenchyma undergo formation of air cavities, right? Large air cavities between the cells, and these air cavities help the plant to give buoyancy, right? Mainly to the aquatic plants, right? So aquatic plants contain parenchyma in large quantities in the plant body that helps the plant float on water. So those are the subtypes of parenchyma tissues. There are many more types, but we are going to learn only these types, right? Whereas the complex tissue, the complex permanent tissues are basically of two types. One is the xylem and the phloem. Both of them are simply termed as vascular tissues, as they form the conducting systems of the plant. Now, xylem actually are the water conducting tissues. The xylem is made up of 4 different types of tissues. That is why they are called complex tissues. Let us see which are those. They are the xylem tracheids, xylem vessels, xylem parenchyma, and xylem fibers. All these help in conducting water from the roots to all the other parts of the plant, whereas the other type called as the phloem are the food conducting tissues. Okay, the food conducting tissues of course are living tissue, except the phloem fibers, which are sclerenchyma fibers associated with phloem. The phloem also phloem actually are food conducting tissues. They transport food from the leaves to other parts of the plant. Not only that, the movement of food materials like sugars and amino acids takes place to and fro throughout the phloem. Okay, the sieve tubes or sieve elements contain the sieve plates, which are very distinguished plates for transport of food. Right, phloem consists of 4 different types of tissues: the sieve elements or the sieve tubes, the companion cells, the phloem parenchyma, and the phloem fibers. In this way, we have seen the different types of plant tissues, right? I have briefed it for you. I hope you recall all the points. Now, today we are going to actually enter into the world of this new field of science called as biotechnology. What exactly is biotechnology? Let us see. The techniques of bringing about improvements in living organisms by artificial genetic changes and by hybridization for the welfare of human beings are together called as biotechnology. Now, last session we had learned about biotechnology, briefing one method of biotechnology, that is tissue culture. Let us just recall all those points once again. Now, biotechnology is a field which has 2 basic methods which are which bring about the improvements in living organisms. They are the tissue culture and the genetic engineering, right? So let us talk about the tissue culture first. We have learnt already that ex vivo growth of cells or the tissues in an aseptic nutrient rich medium is called as tissue culture. With the help of tissue culture technique, a complete organism can be developed from the single cell or from the tissues. Okay, so this was a complete turnover of the technology in the field of science. Anyway, this tissue culture was actually implemented by using a liquid or solid or gel-like nutrient medium which is prepared from agar and supplies all the nutrients and energy for the tissue culture which is being used in this technique. So which is very, very important, right? So let us now focus on the various processes in tissue culture in brief. Now we have seen with an example of banana over here, which is a source plant from which a small bulb corm is being taken up as a source. We take small bits, or we call it as explants, which are being inoculated in the nutrient agar medium after the primary treatment and allowed to grow and multiply in controlled conditions, both physical and light conditions, that is in the tissue culture laboratory. Now, once it starts multiplying, of course, these tissue explants form callus. Now, callus are irregularly grown cells, okay, which has to be differentiated. That can be done by using growth hormones like the plant growth hormones like auxins, gibberellins, and cytokinins, so as to induce shooting as well as rooting in the callus cells. When the root initials and the stem initials or the shoot initials develop, they start developing the roots and the shoots, that is the stem, the leaves, which are being developed in few days. Now, these plantlets, which are grown in the lab conditions, in the control conditions, has to be taken to the natural conditions, transferred to the natural conditions. Now that process is known as the primary hardening process. Okay, that is when the plantlets are actually taken to the, are introduced to the soil like substances, like first the compost is being generally used, and the plantlets are transferred into a compost, slowly taking them outside in the outer environment, in the natural environment. Through secondary hardening process, they are brought to the fields, to the soil, to the natural environment, where they start facing the climatic conditions and of course withstand all the stress, and are ready for living free as the other naturally grown plants. Okay, now this is how the various process of the in the tissue culture is generally carried out. Of course, for various plants, there might be many changes in the chemical procedures or in the lab procedures, but basically this process of tissue culture is followed. Right, so let us talk about the next method of biotechnology, which is genetic engineering. That is making modifications or making changes in the genes, in the genetic material of the organism, of inside, which is present inside the cell of every organism. That is a that has all the characters stored in it, the DNA, right? So when part of DNA, that is genes, are being modified with desired genes for desired characteristics, then this results in plants which are called as genetically modified plants or transgenic plants, right? So genetic engineering is the deliberate modification of the characteristic of an organism by manipulating its genetic material, called as the DNA. So that is how the transgenic plants, the hybrid plants are developed. Let us see the basic process of genetic engineering. The plant part, which will be the source plant used for this process. The plant part is being taken up, and the explants are prepared and inoculated in the nutrient agar medium, but along with the desired gene, which is incorporated in the bacterium, which are these bacteria are actually the vectors of the transfer of desired gene into the explant cells. Now when the explants are being inoculated by even the bacteria are going to make entry into the explants, and the new cells that are developed contains the new desired gene, which is wherein the actual DNA is being modified, is being slightly modified because of the incorporation of the new and desired gene. Now such plants, when the callus develops into new plants and plantlets, these plants are simply termed as transgenic plants. Nowadays lots of transgenic plants are being developed, and of course are in the market for us to eat. So we consume nowadays some of the transgenic plants, and you can see the new plant with the desired character. That is here the color of the leaves are different, shown different. That doesn't it is just a diagrammatic representation. You can see the change in the color of the leaf here, which can be called as the transgenic plant. Okay, so well, that is just a diagrammatic representation, friends. So naturally growing plant can be developed into further in the next generations using the tissue culture technique into a transgenic plants. So that was the basic process of genetic engineering. Of course, for every different plant, different differences in the chemical and the lab processes procedures may be necessary. Okay. Right. So, of course, tissue culture is going to lead to many uses of these crops, many changes has taken place in the development and management of agriculture. Let us see what are the different changes in agricultural management due to biotechnology. In the last session, we have already seen in detail about all these things. Let me just brief it to you. Due to the use, let us start with the changes. Due to the use of the seeds of GM crops, that is genetically modified crops, there's improvement in nutritive value and decrease in the loss of crops. In this way, as seeds of a better quality of crops are being produced, farmers all over the world are cultivating the GM crops on large scale nowadays. High yielding varieties of the genetically modified crops like banana, maize, rice, potato, soybean, tomato, cotton, apple, brinjal, beet, rose, papaya, tobacco, wheat, etc. are available in large quantities. The pest-resistant genes have been introduced in some of the GM crops. Nowadays, we are having all these crops in the market. The BT cotton, the golden rice, the tomatoes, the potatoes and the maize, all these are the pest-resistant crops. We also saw the application of biotechnology in floriculture, nurseries and forestry, wherein the nurseries actually contain different types of plants which are necessary for growing gardens or afforestation and reforestation. The seedlings have to be supplied in large numbers for all these purposes. So these nurseries contain a large number of such plants. It is profitable in such cases to produce plantlets. Now, in this case, nurseries require to produce lots of plantlets by tissue culture techniques. The tissue culture can be actually used for growth of plants on a large scale, which bears beautiful flowers and fruits of excellent quality. Now, fully grown plants can be produced in short duration also. That is, the time taken for the production of plants can be reduced, and not only that, these plants can be grown in large scale on the nutrient medium, that is the tissue culture nutrient medium, by using bioreactors, wherein the GM crop plantlets can be produced on large scale. We also saw that the plants can be grown without, or even if the pollinating or the germinating seeds are not available, because it requires only a tissue, a small group of cells to multiply, and that can be done through tissue culture. For example, plants like orchids, pitcher plants, or some exotic plants can be grown using tissue culture in large quantities without without worrying about the pollinating vectors or even the germinating seeds. Okay, non-availability of all these things. Now, we also saw about the plants that are produced, whether they may be the transgenic plants, most probably are disease free, and these plantlets which are produced by tissue culture, if we use the meristematic tissues, then they are even virus free plants. So these plants, disease free plants, can be grown. Not only that, the time required for the growth of the plants is very, very less. Okay, so we can reduce the time also, and in short time we can produce large number of plants, the required plants. We also saw that the hybridization techniques can be utilized to produce well and successfully grown plantlets, because some plants may undergo some kind of may undergo some kind of um, some kind of internal may face some kind of internal factors wherein the embryos do not form the plants, okay, do not grow properly, and that is why these conventional hybridization techniques between two or more varieties may not grow fully for some reasons. In that case, the embryos produced by tissue culture technique always are successful, are can be grown successfully. Now, some rare and endangered plants also can be grown by tissue culture techniques, okay, and in this manner we can restore the and protect the plants which are endangered from extinction. So, by preserving the tissues of such plants, we can also protect various varieties of those which are rare plants and endangered plants. Okay, so application of biotechnology in floriculture, in the nurseries and forestries, all these are being nowadays taken up very seriously for the conservation of the environment. Let us see the major uses of biotechnology. Biotechnology, of course, the basic methods of biotechnology are being used in the production of cash crops, improvement in varieties of cash crops, increase in the abilities of plants to withstand environmental stresses. The vaccine production nowadays, you know that vaccines are being produced in large quantities for COVID-19. So biotechnological processes are being utilized on large scale to produce vaccines. Early diagnosis of some congenital diseases can also be done through these procedures. Organ transplant, cancer research and production of even artificial organs like skin, cartilage can be taken up in laboratories. So artificial skin and cartilage, of course, are animal tissues. So you must have understood that biotechnology is not only used for plants but also for animals. Using the animal tissues, we can develop various animal organs and maybe the whole human being sometimes in future. Right. So before we let us start with the next topic, actually subtopic, but before we start, let us see how much you remember or how much you can apply you will use your brain power. Come on, let us enjoy this session. Which other industries can be developed as an extension of the plant nursery business? Oh my gosh, plant nursery is a business. Yes, it is a business. And can you try to name other industries which can be developed? Of course, let me tell you, it is already over there on your screens. Agro-complementary occupations like the animal husbandry, poultry farming, sericulture, even many more. You have cattle rearing, you have apiculture. There are many more agro-complementary occupations. Okay, so let us see a few of those, and these industries can be developed as an extension of the plant nursery business. To which places do people generally choose to go on vacation? Yeah, I know you all love to be on vacation, but nowadays you are on vacation already, should I say, because most of the year went being at home, most of you students, in order to relax when they are tired of crowds and a stressful life. So to which places would you love to go? I know you would love to go to either hill stations or to some seashores, or sometimes some of them may choose even going to remote villages, isn't it? Right. So people choose to go to villages, hill stations, seashores in order to relax when they are tired of crowds and a stressful life. Right. That is it. We call that as agri-tourism. If you choose to go to remote village or countryside, of course, it is going to develop a new business, that is agri-tourism. Let us talk about agri-tourism in brief now, so you can think it over as a career also, students, right? So being in remote village also calls up for tourists to visit your place. Remember that. Let us see what is agritourism and what exactly is can be done under this. If sufficient land is available with you, the emerging field of agritourism could be a very good business. The plantlets of flowering, medicinal plants, ornamental plants, vegetable plants, crops, and fruit trees can be produced on large scale by tissue culture techniques. Okay, so you can see here, of course, the strawberries are in up in the market now. Of course, you can see various fruit trees like bananas, you have papayas, you have chicus. There are so many, the guavas, the pomegranates, so many exotic fruit varieties that can be grown if you have a large land available, sufficient land available for the growth of these plants. And you will have to just set up a small tissue culture lab wherein you can start increasing or multiplying the plants using tissue culture technique, okay, if possible, right? Right. And by growing some of the plants fully, a self-sufficient agritourism center can be developed there. Okay. Like you can have shade giving local or exotic attractive plants of flowers and fruits, the ornamental and flowering plants, butterfly garden. Of course, if you have a garden with beautiful flowers, of course, the butterflies, beautiful butterflies, the dragonflies, the bees, all these visit your garden. You can call it as a butterfly garden, and tourists are of course attracted to them. Now, medicinal plant garden also can be utilized, wherein nowadays medicinal plants are in large use in large quantities. You can even supply, you can also enter into trading of these medicinal plants if legally. Not only that, you can have a garden wherein you can talk about the medicinal plants and their uses to the tourists. Now, organic vegetables and fruits. Nowadays, organic farming is a very upcoming farming procedures, wherein the farmers don't use chemical fertilizers, nor pesticides, nor insecticides. So these crops which are grown are simply termed as organic vegetables and fruits. So your place, your remote village can become a very attractive tourist place, okay, under agritourism. So let us now see so agritourism can be a very good business, but you will have to have a good study of all these. Now, next let us go to the agro-complementary occupations. Let us see a few of these agro-complementary occupations. One is the animal husbandry we are going to study in brief, the poultry farming, and the sericulture. Agro-complementary occupations. Under this, let us see animal husbandry. What is animal husbandry actually? The number of cattle, their variety, total milk production, cleanliness in cattle shed, arrangements for health care of cattle, all these things together is called as animal husbandry. Now, in India, animal husbandry is practiced generally for milk production and for using the cattle as help in farming operations. For example, cows and buffaloes are raised for milk, and bulls and male buffaloes for pulling the heavy loads. So, local Indian varieties of cows like Sahiwal, Sindhi, Gir, Lal Kandhari, Devni, Khillari, Dangi, etc. are the various varieties of Indian cows, and exotic varieties like Jersey, Brown, Swiss, Holstein, etc. are kept for their milk. Proper use of cattle is necessary for a clean and high yield of milk from them. So you all have heard about the White Revolution, of course, wherein production of milk was the basic aim, the goal of the White Revolution, which started in India, of course, by Vergis Kurian, and Amul was that big organization that turned the table of production of milk. Right. Anyway, let us talk about how to take care of the cattle if you are dealing with animal husbandry. Health care of the animals under animal husbandry. Basic care should be taken, that is, the animals should be provided with a balanced diet, that is, which includes all the constituents of food which should be given to the cattle. It must include fibre-rich coarse food, the fodder, and sufficient water should be provided. So a balanced diet is necessary, as of course food is necessary for us to lead a very good life, to lead an energetic life. The same way a balanced diet is required even for these cattle. Not only cattle, every other animal. Yes. The cattle shed should be clean and dry with proper ventilation and of course a roof, so that the cattle are feel safe, they are safe from all the germs causing the diseases. Not only that, they can be protected from rain, wind, and of course any other storms that may damage the sheds, okay, cattle sheds. So we should take care of the proper ventilation and roof, and keep the shed always clean and dry. Cattle should be regularly vaccinated, very very important. So vaccination procedures should be followed for cattle also, so that they can be healthy and they can provide us with lots of milk and required products. The next in the list is poultry farming under agribusiness. The rearing of eggs and meat yielding chickens is called poultry farming. The objectives behind the development of new hybrid varieties from a cross between the Indian varieties like Asil and exotic varieties like Leghorns are actually to produce good quality chickens in large numbers. So of course breeds are are in the market nowadays, wherein crossbreeds, wherein Indian varieties bred with the exotic varieties. The Asil is bred with the Leghorns to produce good quality chickens, and of course this objective is being fulfilled nowadays. Well, it also another objective which is very important is to develop the ability to withstand high temperature, to use the byproducts of agriculture as the poultry feed. So agricultural waste, agricultural byproducts can be utilized as poultry feed, and can be, of course, it actually comes under the waste management procedures also. And eco-friendly too, isn't it? So these chickens, eggs and chickens, of course, they develop the ability to withstand stress as well as high temperatures. So the hybrid varieties are also developed in the poultry nowadays. Now let us see which are the varieties of chicken reared for eggs and meat. Rhode Island Red, New Hampshire, Plymouth Rock, Black Rock, all these are varieties of chicken reared. Basically, there are two types of birds. One are the layer birds, and the other one is the broiler bird. Now, layer birds actually are the chickens which are raised for eggs only in the poultry. Where, for example, the Leghorns, the Minorca, the Ankana, and the Layman. All these four different types are generally raised for their eggs, whereas broilers are chickens raised for meat. Okay. The Brahma, Long, the Cochin, and the Asil are the Indian varieties raised for meat generally. Right. So, of course, poultry farming is not as simple as people think, but a lot of work you need to think about the chickens which are raised for eggs, that is the layer birds, as well as the broiler birds which are raised for meat. Next in the list is the sericulture. Of course, this is also another agro-complementary occupation where the silkworms, actually the larval stage of moths, the pupal stage of the moths, we call them silk moths generally, are reared for production of silk. Bombyx mori is the most commonly used variety of moth. These moths lay their eggs on the lower sides of the leaves of the mulberry trees, okay, or the ain trees. So the silkworms are being reared under this business called as sericulture. If you see the life cycle of the silk moth, the adult moth after sexual fertilization undergoes the female moth produces a lots of millions of eggs and deposit those eggs below the under the leaves of the ain trees or the mulberry trees. So thousands of eggs are being deposited, and these eggs after a few days are going to break, and the larvae are going to peep out of those eggs. These larvae start feeding on the mulberry leaves, okay, and grow and develop into large larvae. After a few days, these larvae actually start growing and spinning around themselves a silk thread, okay, and they form what is called as the cocoon, in which the pupa actually develops into an adult moth. The cocoon actually, if you see the cocoon is very shiny, specifically, okay, pale in color, or should I say pale white in color. And of course you can see here a cocoon which is being laid there, which is being kept for you to observe. Now this cocoon may be spherical in shape. 10 days before the pupa turns into an adult, all these cocoons are collected and transferred into boiling water. Why is this done? Of course, these cocoons are the raw material for silk, okay. Due to the boiling water, the pupa inside it dies inside the cocoon itself, and the silk fibers become loose. These fibers are unwound, processed, and reeled, right? Of course, it is a tiring job, but various kinds of fabrics, silk fabrics, is woven from these silk threads. That is how you get silk. Of course, it's a good earning business. Sericulture can be taken up as an agro-complementary occupation. That is it. Let us conclude this session, in which we have learnt about the agritourism and the agribusiness. Under agribusiness, we talked about, in brief, we talked about animal husbandry, poultry farming, and sericulture. Right, let us just see through what we have learned in this lesson number 17, Introduction to Biotechnology. In this, for this chapter, we had 4 live sessions of YouTube, and we learnt about the tissues, the animal tissues as well as the plant tissues were being taught in brief, whereas the tissue culture in plants was briefed to you. Not only that, today we completed this chapter by agritourism, by knowing about agritourism and various agro-complementary business, which are simply termed as agribusiness. So that is all, folks, in this session. We complete this chapter in these 4 sessions, session 1, 2, 3, and 4. I hope you have understood this chapter very well, and Let us meet up later whenever you ask for explanation of any other chapter. Thank you all, and have a good life.